Bismillahirrahmanirrahim. We start the topic-wise discussion on the paper 2 questions which are in the 9700 biology, and this is the AS level exams which you are preparing for. We will do this some questions on protein synthesis to see how well you've understood this chapter. As we start the discussion on question number 1, complete the table to describe 3 differences between DNA replication and DNA transcription. And they've given you a table and you must do a comparative point and then you can get 1 mark for that. So if you have to do a correct comparison, only then you can get a 1 mark. So describe 3 differences. So this is 1, this is 2, and this is 3 differences. So you have to give me 3 differences between DNA replication and DNA transcription. Now this is a diagram which I just managed to get, and you can see how DNA to DNA. So DNA replication is DNA to DNA. The DNA strand is replicated. So from a double-stranded DNA, what we are getting is now 2 semi-conservative replication of the DNA. But in transcription, we have DNA and then we have the RNA. So transcript is the RNA. Transcription, I always say it's a script for a protein molecule. So the word script is in transcription and it is DNA to RNA. And then of course that's always happening in the nucleus. From the DNA is copied by the mRNA, and then the mRNA leaves the nucleus and attaches to the ribosome where proteins are synthesized. Now in this question I'm showing you the mark scheme, and you can see the heading is DNA replication and the heading is DNA transcription. Now look at the point, it says must be comparison statement per row. So the first one is DNA formed, mRNA formed. So always talk about the same thing. Like if I'm comparing two boys and I say he wears glasses, he does not wear glasses. I'm not saying he wears glasses and he is taller. So talk about the same thing. So what is the end result? DNA formed, mRNA formed. Then 2 identical DNA molecules, 1 mRNA molecule formed. And the molecule word is underlined. So you have to use that word. Then product is a double-stranded DNA. Product is a single-stranded mRNA. Then all of the DNA molecule is replicated. Part of a DNA molecule, which is only the gene for a certain protein, which is converted into an mRNA. Both strands are involved. Only one strand is involved, because in DNA, when the DNA unzips, then only one template strand is copied. And then it zips up again, and that mRNA leaves it. Then uses DNA polymerase, uses RNA polymerase. Then DNA nucleotides required, because when DNA is being made, so DNA nucleotides will be required. Here RNA nucleotides will be required. If you don't know that, you need to look it up. What is it? Our DNA nucleotide is deoxyribose. RNA nucleotide has ribose, and the bases, there's the difference, ATGC. In RNA it's AUGC. Now the next comparison was number 8, was process involves complementary base pairing, AT and GC. Here complementary base pairing is AU. The GC part is the same in both. So the differences, because they asked for the differences, they didn't ask for the thing, they didn't say compare. If they say compare, then it's similarities and differences. That's why ignore CG. The story is ignore CG. So if you have to ignore CG, why is there? You must always understand why is that an ignore there, because it's the same thing. CG is in both, in DNA replication and transcription as well. Then takes place in late interphase or S phase of interphase, takes place throughout interphase. Important in cell division, mitosis, meiosis, and this is essential for polypeptide or protein synthesis. So understand these, there were 10 points, you're only supposed to give me 3, so you could have easily got the 3 out of 3. Going to the B part of the question, errors during replication may lead to gene mutations. Errors during replication, when the DNA replicates, maybe a wrong base gets attached. Define the term gene mutation. Change in the sequence of bases. We had A, T, 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 or C, and now instead of this we have A, and we have, instead of T, we have a C here. The rest of it is all the same. So this would be a gene mutation. Define the term gene mutation, because there's another thing which is called chromosome mutation, which an extra chromosome is present. So change in the sequence of bases, change to give a new allele. Allele means an alternative form of a gene, like you have hemoglobin normal and then you have the sickle cell hemoglobin. That's an allele. Change results in non-functioning protein. Yeah, the hemoglobin molecule is not going to function properly. And this results in a different primary structure. So the primary structure of that protein becomes different. Why? Because the amino acid has changed, because the sequence of bases have changed. So that results in a changed amino acid and a different primary structure. Some disease-causing organisms undergo frequent mutation, changing their surface antigens. So this is a virus with this antigen, right? Now this antigen changes. It becomes something like this. So making the disease much more difficult to control with a vaccination program. Explain why existing vaccines may no longer be effective. You see, if previously this was the type of antigen, then these were the type of antibodies that were made. But now that the virus has changed, this requires now a new type of antibody. So this new type of antibody is not going to work because we have got them ready in this type of antibodies. So why existing vaccines may no longer be effective when the surface antigens of a disease causing organism change? Why? Because when you have given them, they are specific. Antigens are specific and antibodies are specific or complementary to that antigen. And the B cells will not recognize this. You have memory B cells. B cells do not recognize this new antigen. Do not recognize the new antigen. So there is no recognition. So there are no plasma B cells and memory B cells. And so they will not make antibodies against this one. So virus, this virus or this disease causing organism will flourish because now the antigen has changed. So the existing memory cells are no longer activated. So specific in the fact that the existing memory cells will no longer be activated and a new immune response is required. Then we come to the part 2 of this question. State precisely the type of immunity. State precisely the type of immunity gained by a person who has been vaccinated. Naturally, it's artificial active immunity. Artificial active immunity. Why active immunity? Because what you gave them weakened viruses. So their lymphocytes performed activity. So and then the memory B cells developed and the plasma B cells made antibodies to kill that weakened virus, which of course was not going to cause any disease. So artificial active immunity. The virus causing measles is said to be antigenically stable as it rarely mutates. Rarely means does not ever. Rare is something very, it doesn't happen, never happens, once in a blue moon. Measles vaccination programs have been successful in preventing epidemics in many areas. Outline 2 reasons why measles is still common in many parts of the world, even though the vaccine is available. Of course, there are many reasons for this. List the reasons: malnourishment, because they don't develop any antibodies, they have protein malnourishment, so no response occurs. Some people do not respond to vaccines. One dose is not always effective. You need to give booster doses, and maybe the booster doses are not, we cannot reach the people who got the first dose, so they do not get the booster dose. Then the cost to authorities, then vaccine may not be thermostable, then overcrowding increases the chances of spread. Number 2, read the following passage. A method called in vitro translation is often used by scientists to produce proteins in the lab. Always read the question very slowly. Used to produce proteins in the lab. The method uses extracts from animal cells, plant cells, or bacteria. The method uses extracts from animal, plant, or bacteria. So it's eukaryotes and prokaryotes. These are chosen because they have high levels of protein synthesis. Like I always say, in your house, maybe you have one chula to make food or to cook a cooking thing, but in a restaurant, you'd have many stoves and many burners to really cook the food. So the cells are treated so that the cell walls, if present, so if it's a plant cell, cell wall, if a bacteria, cell wall, and cell membranes are all broken down. So the cell wall and the cell membranes are all broken down. Now what is left inside? Now the left inside is the cytoplasm with all the stuff in the cytoplasm. So we've broken down the cell wall and the cell membrane. And then, so if present, are broken down and then treated so that any of the cell's own DNA and mRNA, cell's own DNA and mRNA are also destroyed. When mRNA from any other source, mRNA from any other source, so we've taken mRNA from something, somewhere, is added to these extracts, it will be translated into the corresponding protein. So when I added this mRNA to this cytoplasm with the ribosomes in it, then of course this mRNA will translate into this protein molecule, right? So first let's understand the question. Then we go on to the part, the question says A1, explain why the cells are chosen on the basis of their high level of protein synthesis. Because these cells will have a lot of RER. They will have many ribosomes. They will have the presence of the tRNA molecules. And then, of course, ATP will be available for protein synthesis because we know ATP is needed for protein synthesis. Then it says, why did we sort of the cell walls if present in cell membrane need to be broken down? Why? So that the mRNA which we added would not enter through the cell wall and the cell membrane. Now it can reach the ribosomes, and the cell's own DNA and RNA can also be accessed and easier to harvest protein products. We can then get the protein out of it easily. We don't have to go through the cell wall and through the cell membrane, right? So this is how we had to. Then the next part says the cell's own mRNA needs to be destroyed. Why? Because we don't want the cell's own mRNA to make the protein specific for that cell. We want the protein of our choice. We don't want the normal protein. Say, for instance, a cell from the pancreas, it will make insulin. We don't want insulin. We want proteins of our own choice. So only the desired protein is produced. Other proteins need not be produced. Cell proteins may inhibit process. The cell's own protein may inhibit the process, so that would again cause problems for us. Then the last part says mRNA from any source can be translated in any type of extract. Ribosome function is not affected. Mechanism of translation is the same in all cells, prokaryotes and eukaryotes. All types of cells, mRNA is needed for protein synthesis. mRNA only has one role. Like for instance, if I say, okay, what can bread be used for? You can say, well, miss, bread can be used to make sandwiches, bread can be used to make French toast, bread can be used to make cheese toast. So you have a lot of uses for bread, but mRNA has only one role, and that is protein synthesis. And then the genetic code is the same in all the cells. So in every there, the genetic is a universal genetic code. Codons, anticodons. It is the same for every prokaryote and eukaryote. B part of the question, state two differences between the cell structure used in translation in prokaryotes and eukaryotes. Now this again we go back to chapter 1. Different structure of ribosome. The rRNA is slightly different. The other thing is that there are 80S in eukaryotes and 70S in prokaryotes. And then they're attached to RER in the eukaryotes. There is fixed ribosomes and free ribosomes in eukaryotes. But in prokaryotes, there are only free ribosomes. There is no rough endoplasmic reticulum in prokaryotes. Then it says scientists usually find that the method of in vitro translation is less efficient than in vivo. In vivo means in life, which occurs in cells. Suggests a reason of this. Maybe some ribosomes are damaged. Maybe there are lesser number of amino acids available. Maybe you see, they said, if you remember in the question, they said they removed the cell's DNA. So mitochondria has its own DNA as well. So if they've removed even the mitochondrial DNA, then reduced respiration, mitochondria probably won't function either. So this was it was suggest, I always say, are the ones which are really taxing and which can actually, I will tell you all not to do it, because don't waste your time on it. Because the suggest questions are usually the ones which really check how a child can put the knowledge of biology into use. And he or she has to have a very vast knowledge of biology, just hasn't studied from books, but actually likes biology and understands biology. Now coming to question number 3, table 5.1 shows the triplet of bases on the template polynucleotide of DNA for some amino acids. So glutamic acid, which you write just glue, histidine his, leucine leu, proline pro, threonine thr thr, and valine is val. So they're giving you the DNA triplets which code for the same amino acid. So glutamic acid can also be coded by CTT or CTC. You see the third base changes. CTT, CTC. You'll always see that the first, second remain the same. This third one changes. And this results in maybe at times the same amino acid, like CTT, CTC, the same amino acid. Now it says figure 5.1 shows the base sequences in DNA and mRNA for the first 7 amino acids of the beta chain of hemoglobin. Right? Now they've given you the mRNA. The mRNA is CAC. Now what will be the DNA of that? That will be GTG. Go from here back. Then for this one, they've given you the DNA TGA. So what is going to be the mRNA of that? That will be ACU. Because instead of T, there will not be a T here. It shouldn't be ACT. It will be ACU. Why? Because it is mRNA. And mRNA has adenine, uracil, cytosine, and guanine. While DNA has ATGC. I always say remember it at government college. And this is a university government college. Then it says beta chain. So instead of CUG mRNA, which amino acid will it code for? And that is LEU, leucine. This is this one here. Why? Because CUG, this is the DNA. You see, these are the DNAs. So GAC. So if it is got GAC here, this is GAC. So GAC codes for leucine. So this was what you have to figure this out. Then it says use table 5.1 to complete figure 5.1. State the term used to describe the sequence of amino acids in a polypeptide. That's the primary structure. Primary structure is made up of the number and sequence of amino acids. It's made up of peptide bonds only. P primary, P peptide bonds. So primary structure. Coming to the B part of the question, in sickle cell anemia, the amino acid at position 6 in the beta chain is valine and not glutamic acid. Explain how a single change in the DNA triplet for the sixth amino acid of the gene coding for the beta chain leads to the production of a different amino acid sequence. So, I mean, this is something that is we're covering up this chapter. So basically, it's mutation, it's a base substitution, and DNA has CAC as the sixth triplet, mRNA has GUG as the sixth codon. Now, if you see the mark scheme, it's a mutation, it's a base substitution. DNA has CAC as the sixth triplet. So mRNA has GUG as the sixth codon. mRNA has the codons. DNA has CAC as the sixth triplet. Then in translation, so this would be for transcription, this would be for translation. Different tRNA involved. tRNA specific to valine and not glutamic acid. Then anticodon on tRNA has CAC with valine. tRNA brings the incorrect amino acids, valine, to the ribosome. And further detail, if you gave me incorrect amino acid, incorporate into growing polypeptide chain. So there was a lot of mark schemes that you only had to give me 5 points out of these so that you could get your 5 out of 5. So DNA has CAC. Now this was also in the question, you could see that. mRNA will then be GUG. Then the different tRNA involved, because the mRNA dictates which tRNA with which anticodon is going to come and get attached to it, and so it will bring a different amino acid. Coming to last question 4, figure 2.1 shows the base sequence of a DNA triplet code used to produce mRNA. Fill in the corresponding tRNA anticodon in the space provided. So what was given to you was TAC. Now I've told you 2 ways how you did it. You did the TNA, then you did the mRNA, then you did the tRNA. But there are some students who find this difficult because they get confused. They forget to put the U in the RNA. So now I'm giving you another method. First, of course, DNA, TAC, ATG, so have the T's. But then you know the T's have to be converted to U's. So then you convert the T's into U's. So whichever suits you. I mean, I'm giving you the two different methods of first doing it the normal ATGC story, but then figure out what are the RNAs. So in the RNAs, you convert the T's to U's. If you want to find it this way, I mean, just giving you one of the methods of maybe it can help you to understand it. So this would be this part way. Then coming on to the B part of the question, it says more mRNA molecules than tRNA molecules are synthesized in cells suggest a reason for this. Describe the role of ribosomes in protein synthesis. Now, of course, we've done that in a number, but we'll try and do this again as well. What it says more mRNA molecules than tRNA are synthesized suggests a reason mRNA is less stable because it breaks up immediately after it has gone through the ribosomes. While the tRNAs keep on being reused again and again, they keep on bringing the amino acids to make proteins, and any proteins, they are constantly being reused. They don't sort of die after that. They keep on bringing. So if one tRNA has to bring amino acid number 5, well, it will bring amino acid number 5 for any protein which is being made by that cell. So it is used many times. That was a good question, I think. Describe the role of ribosomes in protein synthesis. Number 1, translation occurs there. Number 2, moves along the mRNA. Number 3, the tRNA carrying the amino acid binds to the mRNA. The binding is of the anticodon, which is on the tRNA, and the codon on the mRNA. And then the formation of the peptide bond occurs between the amino acids. And of course, the enzyme used for it is peptidyl transferase. So you gave me a little more detail. This is for 3 marks. So you give me a little more detail so that I can give you your 3 marks and you can do this question properly. That finishes this video on protein synthesis, and I hope these questions have been helpful and you've understood most of this chapter. And if you haven't, please revise it and then go through this video again. And thank you once again for watching.